Welcome back, life science learners, to our revision lesson. We continue to revise meiosis. We've looked at some terms, we've looked at the important concepts. Now let's try and apply our understanding to a few questions. Question 1. The diagrams below represent different phases of meiosis in an organism. It's important that when you have a diagram that you read the heading. The heading often gives you a description of what is happening. And here in this diagram, it tells you different phases of meiosis. Let's recognize the fact that in meiosis, we have different phases, and these may not necessarily be in the correct sequence. Right, so let's move into that. So here we're finding there are 3 diagrams that have been given to you, each with a unique representation of what is happening in these cells. Let's read further into the question. The first part of the question requires us to identify A. Guys, when we look at the diagram, we can clearly see structure A being labeled, and structure A points to this little structure. Guys, we know that chromosomes have chromatids that are held together by what we refer to as the centromere. So structure A is pointing to that protein that holds the chromatids together. After replication has taken place and copies of DNA have been wrapped around histone proteins to form condensed structures, they are held together by these structures which are called centromeres. So structure A is the centromere, and it's important that you're able to recognize that. Let's look at part B, or identify what structure B is. If you look at structure B, guys, it points to a bracket that encloses all of those 2 chromosomes together. When we look at a bracket that collectively wants you to label 2 structures, the context of that would be that it's pointing to a pair of chromosomes. The next bit would be, are they similar in shape and size? Looking at that, yes, they are. So for me, that would be a pair of chromosomes. Because they are similar in shape and size, I often refer to them, and the correct term would be homologous. So B would be a pair of homologous pair, and that's a homologous pair of chromosomes. And so that tells us that you've recognized that they are similar and that there is a pair of them. Structure C. Guys, when we look at structure C, and those are structures that are attached to the centromeres, and that structure are those structures are the spindle fibers that are produced from the centrioles during cell division, and they essentially control the movement of the chromatids or the chromosomes in the cytoplasm, in the nucleoplasm during cell division. Right, so as I mentioned, spindle fibers or spindle threads that you can use to answer them. Question 1.2. Identify the phases represented in diagram 3. So guys, when we look at diagram 3, it's important that we first recognize what does it show. Is it showing you chromosomes or chromatids? And again, it's important that when we look at diagram 3, we can see that there are chromatids indicated. The next question that you've got to ask yourself, are these chromatids? Where are they? So it's important that we recognize that during meiosis 2, that chromosomes are moving apart. Sorry, let me say that again. It's important that we recognize during meiosis 2 that the chromatids are going to be pulled apart. And here we're seeing that the chromatids are being pulled apart. And so clearly this is meiosis 2, and the reason would be that chromatids are being pulled apart, and that rationale would help us to identify that it's anaphase 2. So that diagram there indicates a phase where the chromatids are being pulled apart, and that is in anaphase 2. It's important that you always remember that there is always a number at the end of a phase when we're referring to meiosis. With the exception of interphase, all other phases, prophase would either be prophase 1 or 2. If there is no number, then you're referencing mitosis. So remember that there has to be a number when we're talking about prophase, metaphase, anaphase, and telophase. Right. As I mentioned, that was anaphase 2. Let's move on to 1.3. Write down the numbers of the diagrams to show the sequence in which the phases occur. Now guys, here you're seeing 3 different cells, probably in a sequence, not necessarily showing all the cells in the sequence. So if we were to arrange these in the correct sequence, we would have to understand what is happening in each cell to be able to chronologically place these in the correct order. So when we look at diagram 1, we can see that diagram 1 for me shows you the formation of spindle fibers and centriole splitting. We can see that there are a pair of chromosomes. I often recognize this as part of prophase 1, where chromosomes are now arranged and the spindle fibers forming. So this would be definitely prophase 1, and I'm going to use the acronym P1. Again, that's not what you're allowed to write in your papers. You've got to say that it is prophase 1. Okay? Diagram 2 shows you the meeting of the chromosomes at the equator, and so this would be metaphase 1. And how I know that it's metaphase 1 is, firstly, metaphase is at the equator, and 1 because we see that there are pairs of chromosomes meeting at the equator. And this is going to be anaphase 2, and here we can clearly see that chromatids are being pulled apart. So these diagrams illustrate essentially the process in the following sequence. So we would start with 2, 1, and 3. So again, this would be the initial process of prophase 1, this would be prophase 2, and then finally here you've got anaphase 2. So as I mentioned, here you can see that it's prophase 2, and purely because you've got a single chromatid for each pair, here you've got 2 of them, so that takes us to prophase 1. So that would be, the correct order would be 2, 3, and 1. The next question would be, state one difference between metaphase 1 and metaphase 2. Guys, during metaphase 1, it's important that we recognize that we still have pairs of chromosomes. So when these meet along the equator during metaphase 1, we find that a pair of chromosome meets at the equator. So that's a pair of chromosomes, maternal and paternal pairs at the equator. And that would be metaphase 1. However, in metaphase 2, we need to recognize that we no more have pairs of chromosomes. So we have single chromosomes that are meeting along the equator. And so the difference between metaphase 1 is that there's a pair of chromosomes that have met along the equator, whereas in metaphase 2, you now have single chromosomes, one from each of the pair arranged along the equator. So in a human cell, you would have 23 of these aligned along the equator in metaphase 2. However, in metaphase 1, you will have 23 pairs of them aligned along the equator. And that's the significant difference between metaphase 1 and 2. Right, so as I mentioned, that's the explanation to that question. Guys, the next question talks about the following diagrams. The diagrams below represent different phases of meiosis. Again, these questions often require that you're able to identify unique phases in this process. It's important that you look at the phases and you identify features in each one of them. As we wrap this segment up, I'm going to assist you in identifying the following parts, and let's get into it. So identify part A. And if we look at the diagram, part A again points to this structure here, and that structure again produces the spindle fibers. So I challenge you to go and reflect on what are those structures that are producing the spindle fibers. I'll hold on for a bit until you give me the answer, or think about that. The next one is structure B. Again, structure B points to a bracket that encloses all of that, and clearly that points to a structure which is separated out from its partner or pair, and that would be one single chromosome. And finally, structure C. If you look at structure C, it points to the cell, and it points to the outer cell membrane, and here we find that's the nuclear membrane here, but the outer wall is the cell wall or cell membrane in that case. So as I mentioned, that could be, A could be the centriole or centrosome, B would be the chromosome, and C could be either the cell membrane or the plasma membrane, depending on the type of cell that we see. So guys, that's a wrap for this segment. Trust that you've been able to apply some of the basic identification traits of each phase. Let's get back after the break and tackle some more interesting questions. See you in a bit, have a quick break, and I'll catch you at the end of that.